Welcome to the Brain and Neuropsychology unit. In this video, we'll cover the structure and function of the nervous system, neuron structure and function of the brain, and an introduction to neuropsychology. I've updated the PsychBoost app. Now it has over 500 GCSE flashcards, over 400 multiple choice quiz questions, and over 300 key terms to test your knowledge with. Of course, all based on the information I cover in these videos. I'm so confident you'll like my app. You can use all the features for Paper 1 for free. Only subscribe to Paper 2 if you know you'll find it helpful. You can also follow along with these videos by completing my worksheets, available on Patreon. The structure and function of the nervous system. So let's start by dividing out the human nervous system, the web of nerves that run throughout your body. The central nervous system, or CNS, is where information is processed. And it contains, of course, the brain, where complex processing takes place, but also the spinal cord that transmits information between the brain and the rest of your nervous system. The rest of your nervous system is the peripheral nervous system, or PNS. It's constructed of a network of messenger neurons, either sensory for bringing information to the brain, or motor from the brain back out to the body. The somatic nervous system, or SNS, is what we use when we decide to perform an action. So if you pick up an apple, you're using your somatic system to coordinate your muscles. We call this a voluntary system, as it's under conscious control. You decided to move or to not. What's not voluntary is the autonomic nervous system, or ANS. This is a network of nerves that controls your internal organs and glands. So things like releasing hormones. You can't consciously decide to do those things, so the ANS is an involuntary system. And we break the ANS down into two opposite branches, sympathetic and parasympathetic. The sympathetic branch activates in a stress response, so during fight or flight. And the sympathetic branch wants to increase bodily activity needed for immediate survival, so increasing heart rate, breathing rate, and sweating. But it will slow the activity of processes not needed for immediate survival, like digestion. The opposite parasympathetic branch is activated as a rest response, decreasing those bodily processes and allowing the body to digest food. And normally there's a balance between these two branches called homeostasis. But as I said, the sympathetic branch activates in the fight or flight response, and that's something we need to focus on. So, the brain detects a threat. An area of the brain called the hypothalamus is the area that decides that a threat has been detected, and it will trigger the sympathetic branch of the ANS. That will cause a hormonal response with adrenaline and noradrenaline released into the bloodstream from a gland just above your kidneys called the adrenal glands. The hormones in your blood will now cause the biological processes to change, increasing that heart rate and breathing rate, but also slowing digestive and immune systems. This allows the body to use all of its resources for immediate survival. There's also psychological changes in the fight or flight response, a feeling of panic and high alertness. But when the threat's passed, the body will return to rest. Now, when you think of emotion, you likely think that an emotion is a direct response to something bad happening. There's an interesting theory of emotion called the James-Lange theory that says something very different about the cause of emotion. The theory suggests that it's actually the physiological changes in our body that causes the emotions. So if I was to draw this out as a diagram, we would have the event followed by a biological response and then your brain's interpretation of what that physiological response means, and then the emotion. So in other words, instead of feeling sad because of a negative event and then crying because you're sad, you would actually cry first and then interpret the crying as sadness. Or to link this to the fight or flight response, you see a tiger, your heart rate rushes, you shake and you sweat, and then you interpret all of that as fear. It's an interesting point. And if you spotted a tiger and you didn't have any of the physiological changes in your body, you would probably say that you weren't afraid. So if we can remove the physical changes that are linked to an emotion, the theory suggests we just remove the emotion. And this is possible according to the James-Lange theory, because each emotion has a distinct pattern of physiological responses. Now, the theory does seem to be able to explain how emotions can be controlled by trying to control our physical responses. And it would make sense from an evolutionary perspective that, with a quick response being a matter of life or death, It would make sense for the body to change first, not waiting for a threat to be thought about emotionally. But there are a range of similar body responses to very different emotions. Two examples are fear and attraction. Both can have a quick heart rate and sweating, but they're very different emotions. The theory struggles to explain how our brains decide on which emotions to feel. There are also a wide range of other models of emotion that seem to have a higher face validity, as in they seem to make more sense. The Cannon-Bard theory, for example, suggests that the physical and emotional response happen at the same time, but separately. Also, the James-Lange theory is simplistic, treating all emotions as a result of interpreting biological responses. Neuroscience research suggests that different emotions are processed in very different ways. Brain scans show that complex emotions are processed slowly and deeply across the brain, while simple emotions like fear travel directly to the amygdala, avoiding the cortex for a quick response. Neuron structure and function. So it's likely you've seen an image like this before. It's a drawing of a set of neurons. A neuron is a specialized cell in the body that transmits information electrochemically. There are three different types of neuron in this picture. The blue one is a sensory neuron. This detects information, so pressure or heat as example, and passes that information through the peripheral nervous system to the central nervous system. The relay neuron in pink detects information from the sensory neuron and can pass information to the central nervous system for processing, and it can also pass information to motor neurons. Motor neurons, drawn here in green, detect information from relay neurons and will carry this information to the muscle, making it contract or relax. Together, this collection of neurons is called the reflex arc. It allows reflex actions, the body moving quickly in response to possible danger. Let's look in more detail at the structure of the neurons. Dendrites are the extensions of neurons that detect signals sent from other neurons. The cell body, common in all cells, this contains the genetic information of the nerve cell and controls the cell's functions. The axon is a long extension of the nerve cell that allows it to pass messages on to the other nerve cells. Axons are often, but not always, covered in a myelin sheath. This works as insulation, making the electrical signal or nerve impulse travel faster. Sensory receptors on a sensory neuron are specialized dendrites that detect external stimuli like heat, taste, or light. Motor end plates are at the end of motor neurons, attaching them to the muscle fibers, used to activate muscles. So I've said the neurons communicate electrochemically. We need to understand that process. The important structure is the synapse. This is a drawing labeled up. I'll briefly explain the process so I can show you what the labels are. So, synaptic transmission is simply how a presynaptic neuron, here in blue, converts an electrical signal into a chemical signal that is then detected by a postsynaptic neuron, here in pink. The chemical molecules released by neurons and detected at receptor sites are called neurotransmitters. Neurotransmitters can be excitatory or inhibitory. More on that in a second. Neurotransmitters are released when the electrical signal, known as an action potential, reaches the synapse. Vesicles containing neurotransmitters then merge the cell wall and release the neurotransmitters so they can be detected by the receptors on the postsynaptic cell. But that's not the end of the process. The neurotransmitters need to be taken back into the presynaptic cell. There are transport proteins to actively do this, so that the synapse is ready to fire again. Now, I said I'd say a little bit more about neurotransmitters being excitatory or inhibitory. The electrochemical message will only be sent on if the postsynaptic cell detects the chemical message and then fires. So, some neurotransmitters are excitatory. This means that when they're detected by the postsynaptic cell, they make it more likely to fire, and this is called excitation. But some neurotransmitters, when detected, make the postsynaptic cell less likely to fire, and that's inhibition. Now, these two processes interact, and the postsynaptic cell firing or not depends on the combination of excitatory and inhibitory processes, and this is called summation. Only if the excitatory influences are strong enough will the postsynaptic cell reach a threshold and then fire. Let's finish our discussion on the neuron by considering Hebb's theory of learning and neural growth. Your brain is constructed of many neuronal pathways. These are connections between groups of neurons. Now, Hebb suggests that when we learn, our brain physically changes, creating new neural pathways. The term we give for our brain physically changing in response to experience is neural plasticity. Hebb argues that if we repeatedly use a connection, then that is repeated excitation. That frequent excitation stimulates the connection, making the synaptic terminal grow. Collections or groups of neurons that fire together are called cell assemblies. And when we learn, new neuronal pathways are formed from their synchronous firing. The more neural pathways are used, the stronger they are, but also the more efficient they become. This is a reason a task becomes easier or performance improves the more times the task is repeated. Let's evaluate Hebb's theory. Well, Hebb's theory can be applied to education. The ideas behind growth mindset suggest that improvements in performance are due to increased effort. This is directly supported by Hebb's ideas behind neuronal pathways becoming more efficient with use. There's also direct biological evidence. Observations under the microscope of neurons growing new connections when electrically stimulated. Biological research is performed with precise tools under careful controls, so this is strong evidence for the physical growth of neurons when stimulated. While Hebb's theory can be criticised as being overly reductionist, effective learning is a complex problem and can't be fully explained just by the growth of new synaptic connections. Structure and function of the brain. Now let's start with some classic views of the brain. Here we have a top-down view of the brain, so the brain viewed from above, a view of the brain from the side, and a cross-sectional view of the brain from the front. Most of what you can see in the light pink on the side view of the brain is the largest brain structure, the cerebellum. It's at the top of the brain, and what's obvious from the top-down view is the brain is split into two halves, called the cerebral hemispheres, one on the left and one on the right. If you're interested in what would happen if we cut the connection between the hemispheres, you can check out my A-level video on just that. The two hemispheres are contralateral, meaning each hemisphere senses and controls the opposite side of the body. If you move your right arm, that was because of a signal sent by the left side of your brain. You can see from my cross-sectional drawing of the brain, there is an outside layer to the brain, and we call that the cortex. It's also called grey matter, as it's mostly cell bodies doing the processing of the brain. The inside, or white matter, is mostly axons, and these are the connections between the neurons. The cerebral cortex is wrinkly, so folded, and this is to increase the brain's surface area, allowing more space for processing. Psychologists divide each cerebral hemisphere into 4 lobes. Let's identify each one along with some of its functions. The left temporal lobe understands and produces spoken language. The frontal lobe is involved in conscious thought, planning, memory, and logical tasks like problem solving. It also regulates social behavior like facial expressions. The parietal lobe processes touch sensations from around the body and brings together information from other parts of the brain. At the back of the brain is the occipital lobe. This processes visual information coming from the eyes. Now, not one of the lobes, but a separate structure is the cerebellum, and that's found at the base of the brain. It's responsible for functions like balance and coordination. Now, I've outlined the lobes and some of their functions, so what they're responsible for. Now, this idea that the brain's abilities are not evenly spread across the brain, but are located in particular places, is called localisation of function. And we should be able to identify a few precise areas of the brain that have particular functions. The motor cortex. Now this runs along the top back of the frontal lobe, and it controls voluntary movement. You have one in each hemisphere, and the brain is contralateral, so the motor cortex moves the body on the opposite side. The somatosensory cortex runs along each of the parietal lobes. It detects touch sensation from the opposite side of the body. And the visual cortex, one in each occipital lobe, processing visual information. The auditory cortex, one in each temporal lobe, processes sound information. And specialist language areas. These are only found in the left hemisphere. Broca's area controls speech production, and Wernicke's area is for understanding speech. So, that was a tour of the brain. We'll now look at the work of one of the people who first identified some of those areas, Wilder Penfield, an American-Canadian neurosurgeon. Penfield developed a type of brain surgery called the Montreal procedure as a treatment for epilepsy. This involved opening up the brain while his patients were conscious. Locating the area the epileptic fit started by electrically stimulating various parts of the brain. When he found the area responsible for the epileptic fits, he would destroy that small area. During electrical stimulation process, the patients would report sensations like smells and visions. So we can say that the aim of Penfield's multi-year study was to investigate how the electrical stimulation of the brain's cortex would be experienced by patients. Over the course of Penfield's long career, he was recorded as conducting the Montreal procedure on 1,132 patients, recording the locations he stimulated with the sensations experienced. He found that stimulating the same areas of the brain in different people produced consistent results. If he stimulated the occipital lobe, he would generate visual images. Stimulating the somatosensory region would produce physical sensations. One area of the temporal lobe he was particularly interested in, when stimulated, seemed to produce memories. This is an area he called the interpretive cortex. Penfield concluded from his work that many functions of the brain are localized, and he also concluded that individual memories are stored within the interpretive cortex and can be recalled with stimulation. Now, evaluating Penfield's work, we can agree it was groundbreaking for cognitive neuroscience. Many of his findings from brain surgery were confirmed years later with modern brain scanning techniques like fMRI. So while much of his work has been confirmed, like his maps of the motor and somatosensory cortex, his suggestion that the interpretive cortex contains a perfect recording of a memory and can be fully recovered is seen as a myth. One of the reasons for the weakness of his initial evidence is, of his 1,132 patients, only 520 had their temporal lobe stimulated, and of those, only 40 reported experiences Penfield claimed were recovered memories. And then later research showed 24 of those 40 patients were suffering hallucinations linked to epilepsy. So, not exactly reliable evidence. And an introduction to neuropsychology. So let's start by defining cognitive neuroscience. You'll likely know that cognitive psychologists create theories on how information is processed in the brain. In the memory unit, we discussed the multi-stored model of memory. Neuroscientists investigate the anatomical structures of the brain. So cognitive neuroscientists combine both of these fields, matching the physical structure of the brain with information processing functions. Fundamentally, cognitive neuroscientists explain how the structure and function of the brain is related to our behavior, what we do, and our cognition, what we think. An important aspect of this work is identifying the location of function, so matching an ability to a particular location. The historical way of doing this research was by investigating abnormal people and their brains. If you had someone with damage to the brain, so a change to the structure, and they had a functional difference, then you could assume that the damaged part of the brain was responsible for that function. Some famous examples are Phineas Gage, who had a metal bar shot through his frontal lobe and then developed an aggressive personality, Clive Waring, who due to illness had damage to his hippocampus and then was unable to make any new long-term memories, and Tan, who had damage to a part of the temporal lobe, we now call Broca's area, and could only say Tan. There are problems with using abnormal brains to study brain function. The sample size is often only one person, so they may be unusual in other ways, and we're interested in how a healthy brain functions. Fortunately, we now have a range of scanning techniques that we can use to study the functioning living brain. We're going to look at 3 scanning techniques: CT, PET, and fMRI. All 3 use a similar looking machine, but they use very different technology to produce their images. Starting with a CT scanner. CT stands for computed tomography. These scanners use a series of X-rays to produce a set of detailed black and white cross-sectional images showing the structure of the brain. A CT scan shows a very high level of detail. This gives researchers the ability to identify small, unusual structures. Another advantage is it's cheaper to perform a CT scan than a PET or an fMRI scan. However, there are some serious drawbacks to CT scans. As you can imagine, firing a large number of X-rays at someone's brain is a radiation risk. And while the images of brain structure are sharp, they don't show brain activity. PET scanners stand for positron emission tomography. This scanner is made of a ring of sensors, and these sensors detect gamma rays. These gamma rays are often given off by a radioactive tracer that's injected into the patient's bloodstream. As more active areas of the brain require more blood to function, the researchers can identify more and less active parts of the brain. So we can see the big advantage that PET scanners have over CT scanners is they show the brain's activity, useful for researchers studying brain function. However, while the radioactive tracers have a low level of radioactivity, it's not perfectly safe, so there are only a limited number of scans a patient can have. The last scanner is the fMRI, standing for Functional Magnetic Resonance Imagery. This scanner uses large magnets to measure the difference between oxygenated and deoxygenated blood. As active brain regions need oxygenated blood, these areas show up in an fMRI scan. The advantages of fMRIs are that they show brain activity and they don't have the dangers of using radiation. But the downsides of fMRI scanners is the images are slightly delayed from when the activity actually happens, meaning they have poor temporal resolution. This means it's hard to measure fast mental processes. A classic psychology study using PET scanner was a researcher called Tulving. Tulving wanted to investigate the connection between types of long-term memory and the brain's physical structure. Tulving injected a radioactive gold isotope into the bloodstream of 6 participants, including himself and his wife. This radioactive gold could be detected using the PET scanner. The participants were asked to recall either episodic or semantic long-term memories. What Tulving found with 3 of the participants was a consistent difference in blood flow when recalling semantic or episodic memories. When recalling episodic memories, activity was detected in areas of the frontal and temporal lobes, but activity was detected in the parietal and occipital lobes for semantic memories. So Tulving concluded semantic and episodic memory are separate processes using separate brain regions for processing. The use of an objective scientific measuring device, the PET scanner, under laboratory conditions provided strong evidence for Tulving's theories about long-term memory. These results are unlikely to be affected by researcher bias, extraneous variables, or demand characteristics. We may question the sample used, as only 6 participants is a small sample, and included Tulving and his wife. Also, only 3 of those participants showed consistent results. So the results might not be representative. Those 3 people could have been unusual in how they process long-term memories. We can also question the validity of the task used in the experiment. It's very difficult to focus the mind on just one thought for any length of time. And taking a CT scan takes time. It might have been that participants found themselves getting distracted and then influencing the results. Finally, let's talk about neurological damage. This is when your brain's neurons are in some way destroyed. If that happens, you can lose the function that those neurons were responsible for. There are many ways that neurons can be damaged or destroyed, but 2 ways are strokes, which can happen when a blood vessel bursts in the brain, releasing blood, or a clot blocks the blood supply to the brain, or physical damage. So a direct impact on the brain can cause scarring, a lesion. Physical damage can also be caused by the effects of a disease on the brain. So problems that can be caused by neurological damage will be related to the area that's damaged. So let me explain this. Motor abilities are how we move our body. If the motor cortex is damaged, then this can cause paralysis of the part of the body that section of the motor cortex controls. But keep in mind, the brain is contralateral, so damage to the motor cortex will cause paralysis on the opposite side of the body. Behaviors, the actions that we take, are regulated from a wide range of areas in the brain. 2 examples are the frontal lobe, that controls decision making, and areas of the limbic system that produce emotional responses. Damage to these areas can change behavior. I want to thank all my patrons for their support. They allow me to work part-time and make PsychBoost. And in return, they get extra resources. For example, GCSE patrons get digital worksheets, exam question walkthroughs, and these videos as a set of teaching slides.